So, hi guys, I'm Dr. Venkatachalam, the surgical oncologist, part of the Surgtest team. So, the next 20 minutes to half an hour is what the discussion entirely is going to be about. And this forms one of the building blocks for your oncology career as well as for your entrance examination. Now, the patterns have been changed, it has been revised. What we see as a part of the examining team part of it, the previous year question papers, we've seen a lot of questions being aggregated to this one particular topic of oncology, which consists of a lot of interesting facts. Yeah, when I was going through the recent updates and the literature, I found two important things. Yeah, the history starts with the Dr. Hippocrates, who defined the term the cancer, and he accredited the same term to what appeared like a crab. Initially, people used to say the breast cancer blood vessels were the one which gave the technical term to appearance of a cancer, and it appeared like a crab-like feature. But what technically appears is the tumor cells will be there and they will have pseudopod extensions into the adjacent normal tissues, and their extensions are referred to as the crab-like appearance. This particular feature denotes too many important things which we will be discussing in today's topic. And we all know Virchow, the father of pathology, who gave the term the cancer is a disease of cells. He found out that cancer originated from a group of cells and it is a cellular pathology rather than an organ-based disease. So he clarified and gave a more in-depth knowledge about where the center of origin of disease started with. And this important person, Theodore Boveri, is one important person who acknowledged that chromosomes are the culprits in the cancer cells. So the chromosomes were identified to be the pathognomonic feature in the years of 1914, almost about 100 years ago. He said the pathological part of any cancer cell will be the chromosomes. And it was in 1950 where Oswald Avery, he said the DNA was carrying the genetic material within the chromosomes, and the alterations of the DNA were responsible for what is causing the cancer cells. And we all know, we've been studying it from our 12th standard, the structure of DNA and the helix model was given by Watson and Crick. Why am I particularly putting forward all these things is, there was one MCQ in the year 2018 DNB pattern where they had asked about who identified the genetic material within the chromosomes as a culprit for cancer cells. So this particular question was one of the remote questions which was asked in general surgery pattern of DNB. But now, because NEET has been conducting the exams, he might still continue to ask the same questions for your SS entrance as well. So these two gentlemen, out of which one is no more, Dr. Hanahan is still alive and Weinberg is no more. These two gentlemen are to be appreciated for their endeavors and their identification of what are the hallmarks of a cancer process. So to know what is a cancer, to know what causes a cancer, we need to know the sequence of events which are responsible for it. So when I talk about Hanahan and Weinberg, we go to the year 2000, where he first identified and gave 6 hallmarks. So in the year 2000, he had said there are 6 hallmarks of cancers. So he said initially starting with there will be a sustained proliferative signal, meaning there will be a genetic element which will cause the propagation of or to the start of a cancer causing nature. And as well, there will be an inhibition of the tumor suppressor genes which will normally control the cancer origin. So this is a double plus which will promote cancer, and this will be a negative inhibition of the tumor suppressors which will further enhance and cause malignancy. These two were the two important hallmarks which was proposed in 2000, along with 4 other hallmarks which have been described in the diagram. Later on, in the year 2011, this was expedited and it was made as 8 hallmarks for malignancy, and there were 2 emerging hallmarks of cancer. These are all not important for any other purpose apart from 2022 version, which came last year, August, who concluded that there are almost 10 hallmarks of cancer. So when somebody had asked me in the year 2000, probably 19 or 18, when I had appeared for my entrance examinations, I would have said 8 hallmarks of malignancy and the MCQ would have been right. But now there are 10 hallmarks of cancers, which individually we will be discussing about each and every one of them in the forthcoming slides. And we have 4 emerging hallmarks and enabling characteristics. What are these emerging hallmarks? These are the non-mutational epigenetic reprogramming, the polymorphic microbes, the senescent cells, and unlocking phenotypic plasticity. Plasticity, you all know, the cellular changes and the ability to multiply. So what causes malignancy is loss of plasticity, and they make the cells become more proliferative. And we all know colonic malignancies have been attributed to a lot of primary pathogenic microbes which cause the conducive environment for onset of a malignancy and propagation of cancer causing events. And senescent cells, we all been talking about telomerase, but now the senescent cells will be also reprogrammed, and they will be starting on to cause the cancer causing events as well. So from this point of this particular slide, I would like to put forward 2 important discussions here. So if there is an MCQ about the emerging hallmarks of cancer and they give you all the previous existing hallmarks, you need to be cautious enough to choose which is existing and which are the emerging hallmarks. So this one particular slide will be one of the high yielding facts which can give you few important MCQs in your exams. This is primarily noted only for the NEET pattern, because this type of genetic algorithm of events will not be discussed in depth in usual NEET pattern questions, because it was in the last year 2022 August this update was given. There are higher chance of them asking in the coming exams. So I said there is not one particular characteristic which is sufficient to cause cancer. Out of the 10 characteristics of hallmarks of malignancy, there is not only one particular characteristic which is sufficient, and we do not require almost all the characteristics to cause a malignancy. It is just an imbalance that occurs within the normal body mechanisms which propagates a conducive environment for malignancy. So we will be discussing about what are each and every single particular characteristics and how each of one of them will be going to cause malignancy. Before that, there is a common doubt amongst most of us whether to call a particular disease as a cancer or a tumor. This is very important because the 28th edition Bailey and Love has given a particular clarification about this in one particular paragraph, which was not there in the 27th edition, that tumor is nothing but just a swelling. It does not mean malignancy, and cancer as such is a sequence of events or the process of events or the disease that is an end stage pathological entity when you see into a microscope. We all know Rudolf Virchow, who gave the classical terms for the inflammation, the rubor, the pallor, the calor, and the daller. Along with that, he also mentioned about swelling. That swelling was later on called as tumor. So Bailey and Love 28th edition has given a particular remark and mention about the word tumor, which should be differentiated from cancer. These cannot be used interchangeably for at least exam purpose. So whenever we talk about principles of oncology, we all know these are the 4 terms which come again and again during our discussions. So we need to know what are each and every one of them. So the first thing that we all know is there is something called a normal cellular structure, and the cellular structure will have the genes within their nucleus, and these genes are coded within their DNA element. So we need to know what is a proto-oncogene before knowing what is an oncogene. The proto-oncogene is a normal gene that is present in almost all of us. These genes are responsible for normal cell survival. So can we know what are the types of proto-oncogenes? Yes, they are the normal growth factors, the receptors for your growth factors, your cellular transmembrane structures, intracellular signals, and the last one will be your apoptotic signals. These are all the normal proto-oncogenes. So for you to understand what is an oncogene, we need to know that the proto-oncogenes are the normal cellular content or the normal cellular structures which are responsible for the cell survival, and they will be in the form of growth factors, their receptors, transmembrane signaling molecules, the intracellular signaling structures, and finally the nucleus transformation or the proliferating signals, finally the apoptotic regulating signals. What is an oncogene now? Whenever a proto-oncogene loses its function and gets a mutation in the form of gain of function mutation. This is very specific. I'm giving a special notice here because the gain of function mutation makes a particular growth factor or your growth factor receptor or your transmembrane signals or your intracellular signals to hyperfunction. Whenever you have increased growth factors, the cell becomes autonomous. So when they become autonomous, this we call increased cell survival. So we normally want the normal senescent cells to die and the mutated cells to undergo apoptosis. We do not want any wrong signals to be propagated into the next generation of mutations of cells. Whenever there is a oncogene proliferation, there will be an underlying gain of function of mutation, which will be responsible for the autonomous lineage, making them self-sustaining. They become self-sustaining. This is the first hallmark of cancer. So I repeat again, your normal proto-oncogenes are cells-regulated genes that are responsible for cell survival. They are in the form of growth factors, your receptors, your transmembrane signals, or your intracellular signals. But whenever these cells become hyper-functioning in the form of gain of function mutation, they go in for an autonomous cell survival and they make the cell or the cancer tissues self-sustaining, meaning they will not be subjected to cell death, or when their number decreases, the continuous proliferative signals will be generated by themselves instead of any other external input. Okay, now when we know what is an oncogene, we need to know what is an oncoprotein. By definition, your proteins are encoded by your genes. You have your introns, your exons, your coding and your non-coding regions, everything that is too much genetic for your SS exams. Once you get into your MCH, during your preparation times for your MCH career, you will have to learn all these things. Your oncoproteins is just for a namesake mentioned here. These proteins are the end product of all your oncogenes. If somebody has to ask me an example for all these, yes, I will tell you. Normal, you take an epidermal growth factor receptor, EGFR. This EGFR is normal proto-oncogene in all your skin elements, your hepatocytes, which are responsible for regeneration. There is day-to-day cells turnover, there is cell death, and there is cell proliferation, and you have your cell migrations. These are all encoded by your epidermal growth factor receptors and your epidermal growth factor. These two are your normal proto-oncogenes. Whenever there is a gain of function mutation of this epidermal growth factor receptor, it results in an oncogenic epidermal growth factor, which is example which you see in your breast cancer or your HER2 receptor. HER2 is otherwise called ERBB2. So again, the same thing I repeat. Your normal coding elements within your normal cell is your proto-oncogene. When they undergo genetic transformation or mutation, you refer it to as oncogene, and they are responsible for what is the production of oncoproteins. These proteins will be used for the production of your cellular elements. Your normal oncogene or your cancer cell has to have certain proteins, your ribosomes, your mitochondria. These are all encoded by your oncogenes. So they keep the cell self-sustaining. So now comes your villain to it. What is your tumor suppressor gene? Your tumor suppressor gene normally is an exact opposite to your oncogene. So if you have, as the name suggests, oncogene promotes oncogenesis, your tumor suppressor gene suppresses your oncogenesis. So the normal tumor suppressor genes that have been taught during our undergraduation, post-graduation every day is P53 and your retinoblastoma. These are being implicated in a lot of malignancies, and a lot of MCQs revolve around this. In this entire slide, what I'm trying to enforce is I will be giving you certain examples for oncogenes, proto-oncogenes, and your tumor suppressor genes. Learn it, understand it in a right sense, and understand the mechanism of function and how they act. because once you know the P53 mechanism of action and retinoblastoma mechanism of action, you will not confuse it with a routine oncogene. A common MCQ that is being asked during all your DNBs and your NEET SS pattern questions is, they will give you a few of the options and ask you to pick out which one of the following is not a tumor suppressor gene, or which one of the following is not an oncogene. So this type of a question will definitely be in one of your examinations, either in your NESS exams or in your NEET SS exams. So be sure about which is an oncogene by understanding the underlying concept. Once you know the concept, it becomes a little easier for you to appear and choose your answer. These are all cakewalk questions. Nobody will make mistake. So be very sure about choosing what is your right choice, unless you get confused about what are the types and what is the basic functioning of each of these tumor suppressor genes and their oncogenes. So these particular genes that I've been talking to are responsible for a normal transformation of a normal cell into a malignant cell. So I've taken an example of one of the famous quoted Vogelstein sequence, Fioron-Vogelstein sequence of malignant transformation. This is exactly the sequence of which these are particularly exact genes, the oncogene and your tumor suppressor gene, that are involved in your malignant transformation. I would take one particular example is your P53. We all know this is very simple. It is your tumor suppressor gene. So as the name implies, your tumor suppressor gene has to be inhibited or it has to be lost. So that is being mentioned here. There is loss of P53 for transformation of a late adenoma into a carcinoma. This pathway will require loss of your tumor suppressor gene so that uncontrolled proliferation will be there. There will be no cell regulation. P53 is responsible for two intricate mechanisms. Most important thing for your lifetime, you can remember that. One, it will be responsible for cell death, apoptosis. Number two is your cell cycle repair. If your P53 is lost, imagine you don't have P53 inside your body, there will be no repair of any mutation that occurs day to day in your life. Number two, there will be no cell death. So imagine anything and everything that has been produced will survive for years together. So loss of P53 will result in a late adenoma getting transformed into a carcinoma. This particular slide can also be asked in any of your entrance question. This forms a part of your GA question, your oncology question, or your general onco principles of oncology part of it. You need to know the sequence. Your normal epithelium will be having the villus structure, which will be proliferative to become aberrant proliferative epithelium. So the APC gene that have been implicated, adenomatous polyposis coli gene, that is gene in 5q chromosome, is your proto-oncogene, meaning there will be a gain of function mutation of this particular gene, which will be responsible for aberrant epithelial proliferation. So it is like starting the ignition of a vehicle. It is just giving a impulsive start to it, but what propagates is a sequence of events which will be your KRAS, again an oncogene. Again, there will be gain of function. Anywhere in the body there is an oncogene, there will be only a gain of function mutation. Anywhere in the body there will be a tumor suppressor gene, there will be a loss of function of the tumor suppressor gene. This converts an early adenoma into an intermediate adenoma, KRAS. This forms a part of your intracellular cell signaling. And what converts an intermediate adenoma into a late adenoma is your loss of DCC. What is DCC? Deleted in colon cancer. So this particular loss of tumor suppressor, this is again a tumor suppressor gene. When this tumor suppressor gene is being mutated, or there is a loss of function of it, it will be converting an intermediate adenoma into a late adenoma. And the late adenoma will, again, already discussed, there will be a loss of P53 into causing a carcinoma. And there will be subsequent other mutations. There are genes which are responsible for mutation or metastatic mutations. These impulsive events will be coordinated by a sequence of events. There will be pro-metastatic genes and anti-metastatic genes, which I'll be talking about in the last few slides of today's topic. So these are all, there will be a total imbalance in the structure of a normal cell regulation, and there will be a lot of mutations. It is not one particular mutation that causes a cancer. It is totally impossible. Though people will tell BRCA1 has caused your genetic familial breast cancer, it is not exactly so. BRCA1 is being facilitated by P53. It is being progressed by HER2/neu also. So there will be a cascade of genetic mutations starting from BRCA1. So the igniting factor is always responsible for causing one particular type of a cancer disease. Here, the example that we have taken is a colonic primary and a colonic malignancy, for which we would like to take the example of APC gene. Again, for a breast cancer, the ignition is from your BRCA gene, your BRCA1 protein, which is getting mutated, and they will be responsible for causing both your breast and your ovarian primaries. So whatever ignites is usually called as a culprit, and they will label it to that particular structure. One another example or a similar sequence of event occurs in another GI structure, which is your stomach. There will be a normal mucosa which will be undergoing repetitive damage and stress induced changes, which will cause a chronic gastritis, and this particular chronic gastritis will be converted into an atrophic gastritis. Atrophic gastritis will undergo a metaplasia. The normal columnar epithelium will be replaced by your intestinal type of epithelium. That intestinal type of epithelium will be undergoing dysplasia. That will eventually result in gastric carcinoma. This sequence of events is being always studied and discussed with respect to your intestinal type of gastric cancer. When I'm talking about gastric cancer in the subsequent classes, I will be discussing about it. There are two types of gastric primaries, as we all know during our MS time. We've been talking about intestinal type and diffuse type. So the intestinal type is particularly having this particular sequence of events, not for your diffuse type. This sequence, if somebody can remember, I appreciate it. Get yourself some tap on your back and appreciate yourself. This particular cascade is called Chorea cascade. And this is a favorite MCQ for all your NEET SS pattern question. They will be giving you a structure or a sequence of events based on how a normal H. pylori gastritis or any non-infective chronic gastritis can get transformed into an intestinal type of gastric adenocarcinoma. All these things are explained by what is called as your atrophic gastritis, dysplasia, gastric carcinoma sequence. This is being studied in particular in specific for intestinal type of gastric cancer. There are few questions in chat which I will be taking at the last. One question is for the EGFR example. I will be coming back to it again. The two-hit hypothesis is again the same thing that I've been talking to about for your retinoblastoma, again, which is your tumor suppressor gene. The two-hit hypothesis is again a cascade of event. The first hit, the sporadic type, or the first hit occurs inborn. It can be transformed from one generation to the other generation. The second generation or the subsequent attack or the subsequent type of your retinoblastoma mutation, which occurs because of your environmental insult or your infective causes, will be responsible for the onset of event. So whichever is a primary culprit will be given the underlying mechanism of malignancies of causation. For colonic malignancies, it is a APC gene which ignites and starts it. For BRCA, again, it is going to be your breast cancer. The second underlying mechanism or the hallmark of malignancy here is your evasion of apoptosis. I already discussed it. This is your inhibition of your normal cell death. The first thing that we discussed is your self-sustaining signals from your oncogenes. Number one, it is being corrected. We've understood that. Number two is your evasion of apoptosis. Apoptosis is normal physiological process that occurs day to day inside you, me, everybody that we see in the daily routine activity. The apoptosis means in Greek is like referring to a falling leaf. So what happens here, the Greek literature or everything that has been described is it's a normal physiological response. We've been attending our entrance examinations right from our PG preparation time. one favorite question that occurs every time is the difference between necrosis and apoptosis. Necrosis is pathological, apoptosis is physiological. I stop there. I don't want to talk too much of pathology there because it is not required for your SS exams. That is all being done with your MD and MS. Again, apoptosis, I already told you, the mastermind behind apoptosis is your tumor suppressor genes, especially P53. I again repeat, P53 alone does not do apoptosis. It has got a lot of associated genes that are controlling your apoptosis, of which the mastermind control is with P53 because it regulates a lot of underlying genes, which we have read during our pathology times in the 7th chapter, the neoplasia chapter, which is a very big chapter during our preparation times. This particular P53 will be acting via sequence of lot of genes. Example, your ATM, MDM. These two are the subsequent sequence of events of your P53, which are responsible for your metastasis. ATM is a pro-metastatic gene. MDM2 is responsible for your cellular repair and prevention and of apoptosis. It promotes apoptosis and prevents underlying genetic damage being transformed into the next subsequent cell division. The cancer cells thus will be able to evade apoptosis, and they will be multiplying and continuing the wrong impulse or the wrong signals into the subsequent cell divisions, and they will be causing accumulation of cells, which will be eventually responsible for what is seen as a lump or the swelling from a particular organ or a tissue. Immortality is a third important hallmark. This was attached and was understood based on your Hayflick hypothesis. This favorite question was asked in 2020 entrance examination NEET SS, where Hayflick hypothesis means the even normal, whenever they subjected a mammalian tissue into a cell culture, they found out that there is a finite number of cell multiplication, meaning one particular cell will divide into two, this two will divide into again another two. This number of finite division is usually fixed, meaning there will be 40 to 60 finite divisions only for every particular mammalian tissue. Your human tissue, your mammalian tissues, everything will be multiplying only to a finite number of tissues. But when this finite becomes infinite multiplication, meaning the Hayflick hypothesis is being disrupted, then you have origin of your malignancy or tumor cell proliferation. So how do this happen? For normal, even every single cell will have your centromere and your sister chromatids. These chromatids will be progressively having shortening. The ends will be having telomere shortening. This particular telomeric shortening will be responsible for senescence. Whenever there is no shortening of your telomere, it means it is understood that that particular chromatid can continue to multiply and divide and undergo meiosis, and they will be subjected to subsequent cell divisions, and they will finally eventually continue to multiply without any cell check. This is occurring at the level of molecular level. So this is why it is called, the telomeric shortening is called the molecular clock. Again, there was an NEET SS question in the year 2019. The AIMS question, how it was called that time, there was a question asking which one of the following is called as a molecular clock in oncogenesis. Telomeric shortening is the answer, because this will keep a check on the cellular proliferation and cell division. If there is progressive shortening, the telomeres will be subjected to final senescence. How does the cancer cell prevent telomeric shortening? Is because they produce an enzyme called telomerase, which will prevent progressive shortening. So they use this particular mechanism of preventing the telomere from dividing down or decreasing in length, and it will continue to multiply, and that is how the cells become immortal. So if somebody is asking immortality of cancer cells as an hallmark, what is the underlying mechanism? The answer would be a telomere shortening. Do not say P53. P53 is for apoptosis evasion, and telomerase is responsible for your immortality of your cancer cell. Both are totally different. Please understand the concept before you go on to your understanding the future topics. The most important key element of any malignant transformation is acquiring blood supply to a new proliferating cell. So a normal blood vessel will be supplying a normal cellular process of events. But how does a tumor cell produce its own blood supply? By it will do, most importantly, it will construct its own blood supply, and it will outgrow, make the cells become more aggressive by acquiring your new blood supply. Why does a blood supply be very important during malignant transformation? Because this particular blood supply or the acquiring of new blood vessels will enable the cell to have a pro-oxygenated environment. So here comes your Warburg effect. So there will be a mismatch between the need and the supply, or the demand and the supply, whenever there is uncontrolled cellular proliferation. So for that, the cancer cells will have to achieve their need by redirecting the local blood supply towards themselves, or constructing new blood vessels by themselves. How do they construct new blood vessels? They have this elaboration of your growth factor called VEGFR and VEGF. The receptor and the growth factor both are being elaborated by the tissue microenvironment. Whenever there is an inflammation, there will be a cascade of cytokines that is being secreted. One of the most important cytokine during inflammation will be your VEGFR, which is responsible for rubor, or the redness in normal inflammation. Whatever you say, you have an infection, you have got any local inflammatory pharyngitis, or you have gastritis, anywhere in the body, whenever there is a local inflammation, the local milieu will be producing a lot of cytokines. Most important one will be a VEGF, that will be responsible for construction of new blood vessel. The cancer cells have got this smart idea of identifying and producing the same cytokines for self-sustaining blood vessels production for themselves. So they thought they did not harbor it, or they did not acquire it from the adjacent cells. So this particular VEGF and your VEGF receptors are particularly being elaborated, and we as cancer treating doctors, we treat targeted therapies or molecular therapies by inhibiting these two elements, thereby decreasing the cellular proliferation, and the cell demand keeps increasing, and the underlying cells will undergo necrosis. This is a concept of using bevacizumab as a molecular targeting agent in colonic malignancies, your ovarian cancers, wherever you suspect your high signaling from your local cytokine milieu. And most importantly, as surgeons, why are we bothered about this angiogenic competencies? Because whenever you go operate on a breast cancer, there is increasing size of the disease. It shows that will be increasing blood cell proliferation. So when you go for a MRM, or say you go for a breast conservation surgery, whatever it is, whenever you go into a tumor adjacent plane, there will be a lot of blood vessels. Whenever you go dissect around that, there will be a lot of bleeding. So this is not only occurring at a microscopic level, it does also occur at macroscopic level. This was first identified for your perinodal dissections, your lymph node dissections, for your lymphoma, or for your axillary lymph node dissections, for your breast cancer primary. There was a lot of blood vessel oozing. That is how they identified that there were angiogenic competence, or there will neovascularization that was frequently occurring in your perinodal tissues. Most important question that I anticipate can be asked in their subsequent exams is some numbers. The 1 millimeter cutoff is being given as a range beyond which your tumor tissue cannot sustain without producing new blood vessel. It is not a centimeter, it is a millimeter. I have got a last slide today to discuss about a few numbers that are commonly being asked in the exam. This 1 millimeter cutoff is very important, because this is a minimalistic size beyond which the tumor cell cannot sustain without producing or acquiring new blood supply. Once it has got access to the blood vessel, what does it happen? It tries to reach out to other parts of the body. This sequence of events, which we commonly know is your metastatic disease, or which we evaluate through our staging investigations. You do a PET scan, you do a CT chest, you do a CT abdomen to know the local and the distant spread. This is been acquiring the ability to invade the adjacent structures. Cancer cells have this unusual characteristic of disrespecting anything that comes into their way. This sequence of events is responsible for invasion into your blood vessels as well. They have three important mechanisms to invade into your adjacent structure. The first thing, they increase the interstitial pressure, or the pressure within the particular tissue. This is your normal environment. You have an adjacent blood vessel going. It tries to expand it. It tries to increase the pressure inside this, and the interstitial pressure increases, and that thereby causes increasing chance of facilitating invasion. They will also produce few substances which will degrade your extracellular matrix. So if it's your extracellular matrix, your MMPs, your matrix metalloproteases will be produced, and collagenases will be produced. These substances will destroy the local environment. They will help you, the cancer cells, to invade into the adjacent structures. This is the most common mechanism of local tissue invasion for your rectal malignancies. So the rectal cancer, which invades your adjacent prostate in a male or the vagina in a female, occurs predominantly by elaborating all these extracellular matrix dissolving enzymes, and thereby they go into what is called all your T4B status disease. For a gastric cancer, from the stomach, it will be going into the pancreas. At the same time, it can go into the left lobe of liver. All this local extension is only possible by modes of secretion of certain proteolytic enzymes, which helps in dissolving your extracellular matrix. Finally, they will make the cell become mobile. How does that happening is called? This mechanism is being referred to as outgoing or outside to in signaling. This is being referred in 28th edition of Alien Love. Outside to in signaling, or the outgoing to in signaling, meaning your extracellular matrix elements and the structures in the extracellular matrix will be secreting certain enzymes which will be making the cytoplasm and the nucleus mobile, meaning the cell will become locomotive. I repeat again, increasing the pressure is one mechanism by which it will invade into the adjacent structure. That is how it has been. Example is your phyllodes tumor. You have got adjacent overlying skin pressure necrosis. That is how you differentiate a phyllodes tumor from your LABC. In LABC, when you take out a skin sample and biopsy, you will have tumor cells into the biopsy sample, whereas in a phyllodes tumor, the overlying pressure necrosis will have an overhanging skin, or it will have an overhanging cover, which will prevent or which will try to limit the spread of the disease. So I will be discussing about all these things during a breast cancer class. That will be totally different. What makes a cell become adherent to the adjacent structure is your local interstitial pressure within your tumor tissues that increases. That's the first thing that I want to tell. Number two will be your enzymes that are being secreted by the extracellular matrix that will be dissolving the adjacent extracellular matrix. Again, they will be helping in invasion. And last thing will be your outside to in signaling. This outside to in signaling happens, which helps in making the cell cytoplasm and the nucleus mobile, and hence the cell becomes freely mobile to be migrating into other parts of the body. This is different. Invasion is different from metastasis. Please understand the difference. Invasion and metastasis are totally different. Metastasis is when the cells acquire the access into your blood vessel. Invasion is into your adjacent structure. The adjacent structure, again, you can ask, it can be a blood vessel, it can be a solid organ, it can be a normal skin as well. It doesn't bother about it, but any local invasion will be because of one of these three important points. And now the most important thing that has been asked in the common MCQs is your EMTs and your METs. What is EMT? EMT is epithelial to mesenchymal transition. MET is your mesenchymal to epithelial transition. These two are very important things that has been coming up slowly, and there are molecular epigenetic silencing mechanisms targeting all your EMTs and your METs. Your EMT means your normal epithelial tissue will be converted into a mesenchymal element to promote invasion and metastasis. And again, your mesenchymal element is usually there in your extracellular matrix. Say, for example, this is your tumor, and you have got your extracellular matrix here. What is residing here is your mesenchymal element. What is there here is your epithelial element. There will be a change or a transition from your normal epithelium into a mesenchymal tissue to promote and make themselves accessible to your blood vessels and distant part of the body. The same thing occurs in the reverse when it occurs at the implantation site. Say, for example, a tumor tissue which is arising from the breast will have an EMT occurring at the local site. Once it goes into the lymph node, it has to go inside the lymph node. This is called MET, mesenchymal to epithelial transition, from where it will be transformed from a mesenchymal cell into an epithelial cell and deposit here. Whenever there's a conducive environment, or what you call it as a soil, the seed and soil theory is been that here is a seed, the cell which migrates is your seed, and what deposits here is your soil. If you have a conducive soil or you have a favorable environment, this mesenchymal to epithelial transition will help in cell multiplication and deposit being identified particular area. Not all the cells that undergo epithelial to mesenchymal transition becomes metastasis. What is visible metastasis is those which have got a favorable environment. The soil has to be fertile soil to promote deposition of cells and multiplication of cells. I have been talking about the EMT. EMTs have been generally regulated by all these 5 genes. Your SRC is your sarcoma gene. Your RAS is your intracellular transmembrane signaling elements. Your integrins are responsible for your cellular connections. These are the communications between two different cells. You will have your CAMs, ICAMs, ECAMs. These are all your intracellular adhesion molecules. These are being used as your probable genetic signaling molecules for epithelial to mesenchymal transition. Your WNT and beta catenin, we all know it has been studied along with your colonic malignancy. Again, there your epithelial mesenchymal transition occurs. Your Notch gene in your BCC, basal cell carcinoma, again this is again your EMT gene. Mutation of JOX gene will be causing your basal cell carcinoma. Beta catenin is your colonic carcinoma. Almost all of these 5 genes are commonly implicated for your epithelial to mesenchymal transitions. The next important hallmark would be evading your host immunity. Your tumor cell is always your foreign element. So whenever a body identifies a foreign element, your antigen will be picked up by your antigen presenting cells, which is your dendritic cell here, and they will be presenting it to your T cells. Your T cells will be secreting your cytokines to destroy your antigen. This happens for any normal foreign antigen. Your tumor cell is also your foreign antigen. So this should also be having some elements which will be picked up by your antigen presenting cell, which will be presenting to your T cells. But what happens in your body in a patient with a malignancy is these T cells are unable to join with your antigen presenting cell. They are unable to read the signal which they are showing. Normally, T cells will have your T cell receptors, TCRs, that will be sensing the ligands which are present in your antigen presenting cells. The antigen presenting cell will be your dendritic cell. They will be expressing certain ligands, and the receptors will be there in your T cells. Whenever the T cell is unable to read or identify the ligand, the antigen presentation is becoming useless, meaning immunity is being evaded. The host is unable to identify the foreign tumor cell as non-self. So that is very important. Body has to identify and differentiate self and non-self. This is not possible. That is why your tumor cells are at leisure, they are unable to enjoy and proliferate and multiply. This is exactly opposite to your autoimmunity, where your normal cell itself is being identified as an antigen cell. That is being different. So exact opposite of your normal autoimmunity is your carcinoma or a cancer pathogenesis, where the tumor cells are unable to identify a wrong thing as a wrong material. It does not sense that there is a new person who's sitting there. Why I have particularly put this particular person in red is because this was asked in the year 2019. Paul Ehrlich was implicated in identifying the tumor immunogenesis. He was a father who gave the concept of tumor immunogenesis. He is also the great person who's called as a father of chemotherapy. This was later on revisited by both McFarlane and Lewis Thomas, who gave the further theories and postulates to substantiate what Ehrlich had proposed, and it was being accepted. So when I talk about McFarlane, you should also be thinking about some adrenal tumors, adrenal tumor staging. Yes, when I talk about adrenal cancer, I'll be talking about McFarlane staging. This is very important. He has been implicated two important things. Your MCQs can still be there also. What happens here, as I said, the brakes are not properly applied. Hence, it is not been identified as a foreign agent, and body continues to allow it to multiply n number of times. The suppression of tumor associated antigens are altering your immune surveillance mechanisms are the two mechanisms by which the body is unable to identify a tumor cell as non-self. Okay, I repeat, the first thing is your suppression of your tumor suppressor antigens or a tumor related antigens. This antigen will be masked by certain elements which are produced by the tumor themselves. So they will be making them become disguised, or they will be protected from the host immunity. And the second thing is you alter your immune surveillance mechanisms, whereby the non-self is not being identified as non-self. It has been still identified as a normal tissue which has been there in the particular person. So why is it significant for us? Why should we be learning about this? Why should there be MCQs about it? Yes, these are all coming up in the recent times. We've been talking about surgery as a treatment modality of malignancy. Later on, chemotherapy came, then radiation came, and there were innovations in radiations like IMRT, then they came SBRT. All modalities of treatments were being discussed. And now, there is important treatment called immunotherapy with almost all solid malignancies. So this is one of the fourth important branch of oncology that has been coming up. This is being studied extensively only based on this particular hypothesis of tumor cell immunity. I have a beautiful example. There is a normal tumor cell which will be expressing a ligand which has to be picked up by your T cell. The T cell has to signal the particular ligand to identify it as a non-self. But how does tumor cell evade the immunity is by it also produces a new ligand which are present in almost all normal cells. This is like an identity card for a particular college. Say for example, if you are from a particular medical college and if you have an identity card, you'll be let inside your campus very freely. Nobody will be questioning you. The same thing happens here. The tumor cells have an identity card of PD-L1. When they have a PD-L1, the T cell will also have your PD-1 receptor. So the ligand and the receptor will match and sit together, and the T cell will say, yes, boss, you can proceed, you can move around inside the body freely. We are not going to control. So this production of PD-L1 by your tumor cell is the mechanism of host immunity. This is how the tumor antigens are being suppressed or not being identified as a foreign agent. Now, we have a wonderful drug, the immunotherapy drug, nivolumab. You also have come across one common another drug called pembrolizumab. These two drugs are PD-1 inhibitors. What happens? You go inhibit this competitive. This is again a competitive inhibition, meaning that it is a non-covalent bonding. You go make the nivolumab sit inside the receptor where the L1 has to normally sit up. Now there is no space for PD-L1 to be sitting inside. Now this ligand is being expressed. The T cell is free. The T cell will go and inhibit the tumor cell. This is the mechanism of functioning of all your immunotherapy agents. This is one of the mechanism. The other mechanism is your CTLA4. CTLA is again a ligand which is being expressed by your antigen presenting cells. And when you have this particular inhibitor in your T cells, the T cells will be making the tumor cells identified as a foreign agent and thereby destroying them. I again repeat, the PD-L1 molecule will be secreted by the tumor cell as an identity card or a badge to show themselves that they are a part of the institution, or that they are part of the normal system of the body. The T cells will be identifying any cell with PD-L1 as normal, so they will prevent them from being destroyed. What happens when you give nivolumab is there will be an inhibition of the receptor on the T cell which normally binds to your PD-L1. So PD-L1 is not being bound, so the cell does not identify it as a normal tissue, and there will be tissue destruction. The same mechanism, same theory happens for CTLA4. I'm not being too descriptive here because I don't want to take your time. Just understand the core concept. Again, you see here you have a dendritic cell, which is your antigen presenting cell, major histocompatibility complex. This is your T cell, which will have the T cell receptor or TCRs. They will be having the ligand expression site where this antigen dendritic cell or the antigen presenting cell will go attach. Once it gets attached, there will be co-activation. This is very important. This co-activation is by a lot of ligands, not only these, but these two are commonly being implicated in tumor immunogenesis. That is why they have been taking this particular photo for explanation. PD-L1 is being secreted by a normal antigen presenting cell, and which will be sitting onto PD-1, and thereby there is no tissue destruction. The same thing the body, the tumor cell has picked up, and they made themselves resemble the dendritic cell by expressing PD-L1 and PD-L2. Both will be binding to the same ligand. When there is a treatment using nivolumab, there is an inhibitor which sits in between your ligand and your receptor. Thereby, the tumor cell is identified as non-self and it undergoes destruction. So, immunotherapy is based mainly on T cell regulation. This is very important. It is not B cell. I repeat again. For all immunotherapy treatments to work, you should have a normal fighting T cell mechanism inside your body. What our immunotherapy drug is doing is it goes and attaches to the T cell, not the tumor cells. Again, I'm being very specific. They go sit into your tumor's T cells, not the tumor cells, and thereby preventing the tumor cell being covered or being protected and enabling them to a lot of host mediated tissue destruction. Okay. I have got a few questions on the chat box. I'll try to complete the presentation. I'll explain everything again. Even if you have a query on something which have already been discussed, if you want to come again, I'll come back to it. The earlier thing that I've already discussed along with invasion is your metastasis. Now, what happens is you should have a favorable soil or a conducive environment or a favorable internal milieu for your cells to settle down, proliferate, and multiply. This was given by Paget's, the person who described the Paget's disease of the nipple or the in situ malignancy of the nipple areola complex of the breast. Is the same person who described the seed and soil hypothesis. The viable metastasis does not occur just by that. It has to have a favorable soil for it to get deposited, and there will be mutual secretion. Say, for example, you have got a gastric cancer, you have got a lung tissue for metastasis. How does your gastric cancer deposit into the lung? There will be a lot of factors which are secreted, which will destroy the basement membrane, get access to the blood vessel. From the blood vessel, it has to travel into the systemic hematogenous axis, and it has to go deposit into the lung tissue. Just by mere deposition does not mean it will develop into a viable metastasis. That is the difference I'm telling. Just because it is being put down there, it doesn't get an environment there. Again, there will be mesenchymal to epithelial transition occurring there. There will be a favorable internal milieu that will enable, meaning the host tissue or the lung parenchyma has to have a receptor, or it should secrete certain genetic growth factors for these cells to sit down, multiply, and proliferate and develop into a metastasis. So a viable metastasis requires a lot of sequence of events for a deposited cell to multiply and be visible in your imaging studies. Now, what happens for a locally aggressive tumor? This is called your invasion. Your basal cell carcinoma, your giant cell tumor of your bones, all these are based on the concept of invasion, where again, three important things that I've already told you. One, the first thing would be your increasing your interstitial pressure. Number two will be there will be an enzyme, the proteolytic enzymes, the collagenases will be secreted, which will destroy your local environment or the extracellular matrix, and that will enable them into a adjacent structure, and they will be destroying your, say for example, BCC, there will be epidermis, the layer 5 layers of epidermis, there is dermis, there will be 2 layers, papillary and reticular dermis, and you have subcutaneous tissue. For a basal cell carcinoma which originates in the basal layer of your epidermis, will have to locally invade by secreting proteolytic enzymes, proteases. This along with your epithelial mesenchymal transition genes, which I already told you, the Notch gene, will be helping them to acquire access into your papillary dermis, your reticular dermis. From there, they will be creeping some more down into your subcutaneous tissue. This makes them go locally aggressive. That is all attributed to this one particular factor which has been elaborated or which has been secreted by your tumors themselves, the proteases. Most important thing, I told you, the viable metastasis occurs when there is a favorable environment. When there is no favorable environment, they continue to circulate inside the body, and these are called as circulating tumor cells, and this is called disseminated tumor cells. These two are coming up very important because they are prognostic factors for solid malignancies. Example, your breast cancer. Measuring the amount of circulating T cells or the amount of disseminated tumor cells will be useful for prognostic staging of your any solid malignancy. Your circulating T cells, when they are present in more than 5 or more than 7 for respective for breast and colonic primaries, they are highly poor prognostic. Presence of a circulating tumor cell by itself means the patient has got a very bad aggressive disease which you have identified only as a stage 4 tumor. And again, the disseminated tumor cells are not M1 diseases. They are present based on the location within every particular organ. If the same is found within a node, then it is called microinvasive, NMI. If they are present in a bone from a breast cancer, then it is called M1. So there's a difference. It does not always mean your circulating cells and your disseminated cells are same. There's a lot of difference between the two. Circulating cells, by nature, they will be just moving around in your bloodstream. They are poor prognostic factors. Disseminated tumor cells are two types. One, either they can get deposited into any other metastatic sites, your lung, your breast, bone, your brain, liver, for whatever matter, wherever the contralateral breast or skin, wherever. It can also occur into the local draining lymph nodes, where it is referred to as NMI, microscopic disease, or M1 disease, based on the location. The sequence of metastasis is what I already discussed. There is, I already told you, there are metastatic promoting genes and metastatic suppressing genes. Metastatic promoting genes will be your ATM, the most important metastatic promoting gene. Why am I mentioning ATM is because this was one of the MCQ in the year 2021, where which one of the following is not a metastatic suppressor gene was asked, and ATM was one of the options. So this is a pro-metastatic gene. It promotes metastasis and it enables the tissues to spread to other parts of the body. On the corollary, your NM31, the most important metastasis suppressor gene is your NM23. The KISS, BRMS1, MKK4, and Ki1 are the other genes which have been recently studied. This is an update which has been given in Bevita, the latest edition, which has not been mentioned in the previous edition. The previous editions had mentioned about NM23. The other genes, BRMS1, MKK4, have been added to the literature in the latest Bevita. Coming to tumor inflammation, we've talked about tumor immunity and tumor inflammation. Both go hand in hand and parallel. What happens? Your tumor cells will secrete certain cytokines. That cytokines will be identified by the body as inflammatory mediators, and there will be local milieu changes. That milieu changes will be enabling the cells to multiply and divide very easily. The cytokines can be a growth factor. The cytokine can be a growth factor receptor. The cytokine can be an intracellular signaling molecule. The cytokine can be an angiogenic promoter. Angiogenic promoter example would be your VEGF. It can help in extracellular matrix deposition by FGF, fibroblast growth factor. So all these molecules are responsible for your tumor inflammation. And most commonly, your anti-apoptotic factors will be your BCL genes, your BCX genes, and almost. And reactive oxygen species will be produced by your local inflammation. This reactive oxygen species is the primary culprit which will be enabling further additional mutation. If you remember in my previous slides, I would have told you APC is an initiating factor where a lot of other mutations of your DCC, P53, loss of function mutations and all will be there. How does all these been acquired in the person who has an APC gene mutation? It is by production of reactive oxygen species, which will be destroying your DNA. And this particular DNA damage will not be rectified or corrected because already one of the tumor suppressor gene, your APC gene, is being mutated. So further new mutations will occur. And this sequence of events will promote conversion of a normal epithelium into an aberrant epithelium, followed by an adenoma, early, intermediate, and late, finally a carcinoma. So one tumor suppressor gene which is being mutated will make the cell fail to repair the normal genetic damage. That is how further mutations are acquired. If that particular mutation occurs in the coding sequence of that particular gene, 17p, it will help in production of new mutations, which will enable the cancer cell to become more self-sustaining. So any cell that loses its normal function because of uncontrolled proliferation, there's no regulated proliferation, unregulated and uncontrolled proliferation. So whenever they become unregulated, the normal cellular function is lost. For example, any adenomatous cell or an adenocarcinoma producing cell should produce secreting function. Whenever there is a poorly differentiated adenocarcinoma, the cell loses its function completely and there will be no secretion compared to a cell with a well differentiated adenocarcinoma. That is how we've been talking about all the gradings. There is a poorly differentiated, well differentiated, and moderately differentiated based on the resemblance to the normal structure, and physiologically it will give you us an idea whether the cells are functioning or not. It doesn't bother for us to whether it is going to be functioning or not, because cancer is a cancer. We'll have to remove. Whether it is well differentiated, we have to remove, or if it is poorly differentiated also, we have to remove. But why it has been significant is because for a well differentiated cancer, the rate of multiplication is little slower and they will be not responding to your systemic chemo and radiations, where the role of surgery is being upheld. Example will be your appendiceal adenocarcinomas or low-grade mucinous adenomatous lesions of your appendix, where there is no role of chemotherapy. Just you do a complete excision in the form of limited excision or right hemicolectomy, whichever you do, followed by keeping the patient on close observation and follow-up. There's no role of much chemotherapies or radiation that is going to work for a person who's got a low-grade or a well-differentiated tumor. On the corollary, if the patient has got a signet ring cell carcinoma of stomach, that again is a very highly aggressive tumor which has got a loss of normal resemblance to the normal secreting fundic glands or normal secretory functions of your gastric mucosa, where your surgery will be futile if there is no proper adjuvant treatment or there is no proper perioperative treatment. I'm being very clear because the recent trends for management of gastric cancer more than T1B stage is shifting to perioperative chemo rather than post-operative adjuvant chemotherapy. So this is how we've been discussing. And if at all you are unable to give a patient with a strong adjuvant treatment, your surgery is almost equivalent of doing nothing. Your surgery is futile. I repeat again, a cancer cell, when it undergoes multiplication, there will be a grades of differentiation amongst themselves. A poorly differentiated tumor will have no resemblance to the normal counterpart of the cell of origin, and they will not be having normal physiological function of the same thing, which is a blessing in disguise, because those tissues will have uncontrolled proliferation, and their proliferative marker, which is being measured by Ki67, will be too high. It will be more than 70% value, which will enable them to have response to a good chemotherapy agent or a very aggressive radiation. Last thing would be a cellular metabolism or the Warburg effect. I already told the cancer cells have a precarious or sporadic blood supply. So they have an internal relative hypoxic environment within themselves. So they get adapted to it. This adaptation is called Warburg effect. Normal cell will have aerobic glycolysis, where the glucose is being metabolized into pyruvate, and it will be subjected to your citric acid cycle pathway, and the end product will be production of CO2 and ATPs. On the corollary, in malignant cells, the normal glucose is being broken down into pyruvate. This pyruvate gets converted into lactate. This lactate is being used by your adjacent tumor cell for their energy and their metabolism, and they will be subjecting them directly as an intermediate of your citric acid cycle and producing ATPs directly. Instead of undergoing a normal aerobic glycolytic pathway, the cancer cells will have an altered internal metabolism or a dysregulated cellular metabolism in the form of Warburg effect, which is a kind of an adaptation to the prolonged phases of relative hypoxia, which will help the conversion of glucose into pyruvate, which later on gets converted into lactate. This lactate is being absorbed by the adjacent cancer cell for a production of enzyme, their own metabolism. This occurs even in normal individuals. This is subjected during your skeletal muscle energy metabolisms during exercises. So this is a normal event which occurs even in every person. Whenever there is exertion, whenever there is increased demand, the skeletal muscle will be using this pyruvate and the lactate. This Cori cycle will still continue, and that will be regulated by the liver ultimately. But in the contrary, for a malignant cell to have self-sustaining cellular metabolism, they have got an adaptive phenomenon to combat their prolonged phase of hypoxia in the form of Warburg effect. What is the end product of Warburg effect? This is an MCQ which has been asked in the year 2020. Lactate is the answer. This was a final end product of your Warburg effect in your malignant cells. As I told you, the lactate is used for your adjacent cancer cells for their own metabolism, and it acts as a reserve for self-sustaining. I told you this occurs in the skeletal muscles during normal exercise and strenuous activities also. So this particular growth curve was being asked in one time during AIMS 2018 exam. This growth curve is called as your Gompertzian growth curve. This growth curve is to measure the doubling time of your cancer cells. So 10 power 2, 10 power 4, 10 power 6 measures your cells population, and this is your time in specific doubling time. Why I have taken this is because until the clinical detection, which occurs at 10 power 9, your cells will not be seen to your macroscopic naked eye examination or your examination by your staging investigations. Until then, the cells will be multiplying very rampantly, and it will be exponential growth phase. Once they achieve this exponential growth phase and they reach the cells proliferation population of around 10 power 9 cells, there will be a plateau phase. This plateau phase occurs because there is an increase in demand and decrease in supply. So the cell blood supply is very less. There's increased nutritional demand and oxygen demand by the tumor cells, which is not being combated. So the cells will undergo death. So there is no exponential growth. Initially, when the demand was less and blood supply was more, there was an exponential growth phase. Once it achieves 10 power 9, the ceiling phase or the cutoff phase, after which there was no further multiplication, or the multiplication was not as aggressive as it was before, because it is attained a saturation, because there is increased cell turnover in the form of cell death as well, because there is increased demand in the form of nutritional requirement for the cellular metabolisms. The same thing what I've described, the Gompertzian growth curve is for your tumor doubling. It is not only for your normal time measurements of how the cell multiplies, how fast it multiplies until it has a clinical detection, and how it attains a plateau phase after a detection of your multiplication of cells. So as I told you, there's some small max for all of you who are interested in mathematics part of it. For a cell which is 1 centimeter or a tissue which is 1 centimeter in size, there are underlying 10 to the power of 9 number of cells inside it. It takes around 30 generations for the cells to multiply to reach 10 power 9. So for 1 centimeter tumor tissue, it requires 30 subsequent cell divisions to occur to become 1 centimeter in size. This is a favorite MCQ. This was asked in 2017 AIMS, where how many generations are required for a 1 centimeter tumor cell tissue to be identified in the human system was asked. Again, it acquires another 15, meaning 30 to more than 15 for it to multiply and increasing in size to become a fatal tissue which can be having a microscopic metastatic disease at the time of diagnosis. This requires almost around 45 generations of cell proliferation. So the last few slides of the discussion, on the left, what is there is the 27th edition of Bailey and Love, the summary of the malignant transformation of what we've been talking now. And this one is your 28th edition of Bailey and Love. There are few minute changes that has been described here. The last, but not the least, the last two have been removed. Jettisons excess baggage and subvert communication to and from the cellular environment, which I've been talking to as outside in signaling. And jettisons excess baggage is loss of normal cell function. So whenever there is excess of normal multiplication of cells, they lose the resemblance to the underlying normal epithelium and from which they arise. So they lose the ability to multiply or ability to function. If it is your adenocarcinoma, those poorly differentiated tumors do not secrete compared to those of the normal well-differentiated counterparts, which resembles the secretory function of the underlying mechanism of cells. So these two have been removed in the 28th edition, meaning to say that this has not got that much implication for cellular metabolism compared to other established key features of your malignant transformation. So this slide was an interesting slide where I wanted to emphasize why are we learning about this hallmarks, because each and every single hallmark can be targeted for treatment. Starting the self-sustaining signaling molecule are being mutations are responsible for causing cell cycle. Your growth factor inhibitors can be given. Example is your Herceptin. Herceptin is your HER2/neu inhibitor, which will inhibit your HER2/neu proliferating cells, which is beneficial for tumor control. Evading growth suppressors, your P53s are being suppressed. Your epigenetic alterations or epigenetic treatment or epigenetics have a new hold. It has been under research to identify how to promote the silenced tumor suppressor gene to become more active again. So this particular mechanism of functioning is being described as a modality of treatment for cancers. And again, avoiding immune destruction, you can make your T cells engineered. You can make them become more specific to your tumor cells by sensitizing them ex vivo. Outside the body, they can be primed and sensitized to tumor antigens, and they can be injected into it. And that will be identified easily inside the body by the colleague or the other T cells, and they will be destroying your tumor cells. This particular treatment is called CAR T therapy. CAR T cell therapy. This is CAR therapy, which is being implicated as one of the treatment modalities for this, making the cells susceptible to the host immune system. Replicative immortality, I told telomere is important for immortality. When I talk about growth suppressors, I talk about apoptosis. When it is immortality, I talk about telomere length. So there is something called telomere vaccine, telomerase vaccine, which will go and destroy the cancer cells which will be producing your telomerase enzyme. So it is more or like an antibody against your telomerase. Thereby, the progressive shortening can be accelerated and the cells will be subjected to senescence. Growth promoting inflammation, again epigenetic editing and molecular cytological or cytokine mediated changes have been studied under research to promote cell death. And invasion and metastasis, you can identify early presence of metastatic genes and you can inform your patients whether he or she is at a risk of getting a metastatic recurrence at a later age. By identifying a screening candidate, you can keep the patient on more aggressive follow-up if the patient has got a particular genetic alteration which is responsible for metastasis. Angiogenesis, I've talked enough about it. We've been talking about growth factors and growth factor receptors. Even now, there are new researches that are undergoing for identifying microRNA levels, genetic changes that can be done to prevent or to inhibit your tumor angiogenesis. Genomic instability and mutation, you can have barcodings and monitoring of your heterogeneity of tumors, and you can tell whether the patient is going to acquire any further mutation in the recent or the remote past, which will make them getting converted from an adenoma into a carcinoma. If you remember the Vogelstein's model, where I had mentioned initial few mutations, which will be finally being transformed into a final metastatic disease, the acquirance of those particular mutations are possible or not can be identified by this barcoding monitoring. Again, this is under research level. And resisting your cell death or preventing the cell from cell death can be aggravated or it can be accentuated by, again, the same CAR T therapy, which is being described using viral genes, where there is hybrid of genes between the host genes, T cells, and the genes from the viral oncogenes will be incorporated into it and they will be injected. This particular treatment is used for melanoma, where they inject your hybrid cellular components which will contain the host T cells which are remodified with viral oncogenes that will inhibit your anti-apoptotic factors and make the cell into go for a permanent apoptosis. Finally, the last one would be deregulating the cellular energetics. We've been talking about Warburg effect. We've been using the same effect for your PET scans. All your PET CTs or your molecular studies that are being done are based on the Warburg effect. And recently, apart from Warburg effect, another cellular metabolism that is being asked as an MCQ is your FGF22. FGF21 and FGF22. These two are epigenetic changes and the cellular metabolic dysregulating agents which have been implicated as one of the reasons for self-sustainability of tumor cells. FGF stands for fibroblast growth factor 21 and 22. Both of them are being identified as novel agents or agents which are giving hint about a dysregulated intracellular microbial environment or your tumor microenvironment. Both happens when it is from your microbe, polymicrobes that are being identified for your colonic cancers. When there are such changes which are noted into the microbes, they can be identified and they can be corrected and thereby preventing them becoming a final adenoma or a carcinoma sequence. So in short, to remember a mnemonic to remember all the hallmarks will be S, which stands for self-sustainability of growth signals. A stands for angiogenesis. Another A stands for apoptosis avoidance. I stands for invasion and metastasis. Another I stands for insensitivity to growth inhibitory signals. And L stands for limitless replicative potential. This is your immortality, telomerase and telomere. Your doubts, I would like to take all your chat questions first. If anybody has got any questions, you can just keep putting off in your chat box. Once I finish, I unmute all of you. If somebody has got a question, you can ask them. And my number is being displayed. If anybody has got a question who wants to ask privately, you can WhatsApp or call and get your doubts clarified. I go from the last. There's one question asking about loss of specialist function is called jettison's excess baggage. Yes, it is right. Jettison's excess baggage means a normal cellular event is not, or normal cellular function is not happening in a tumor environment. So the normal cellular properties are not being carried to the tumor cells. They just multiply madlessly. They do not have any physiological resemblance or function of a normal cell. That is what is being referred to as jettison's excess baggage. It is useless. And there's a question regarding ICAMs and ECAMs. These are cellular adhesion molecules. These molecules, I already told you, there are integrins. These integrins are responsible for your cell to cell adhesions, and your adhesion molecules are your ECAMs and your ICAMs. These are normally being present on the extracellular surface of your cells. When they have got a genetic signaling between themselves, they will be kept in close adhesion. Whenever there is a mutation of your integrins or your ECAMs or your ICAMs or your VCAMs, whatever happens is this intracellular or intercellular to cellular intercellular signaling is being lost, and the cells become independent, and mobile cells, they can go into access for local tissue invasion, or they can get access into the bloodstream to become metastatic. Okay, there's a question about hypomethylation and hypermethylation rules. I was talking about in my last few slides about epigenetics. Epigenetics is altering your genomic function. How do you alter your genomic function? You make the cell become functionable by making it hypomethylated. I will be talking about this when I'm talking about your colonic cancers. There will be a class for colonic malignancies. When there is an update, I will be talking about it. Hypomethylation makes the cell proliferate. So what we want to do is you make the cells become hypermethylated, or you make the environment or the gene become hypermethylated. There are two epigenetic modifications that are being implicated as treatment options. One is your acetylation or acylation. They are interchangeably used. Another one is your methylation. HDAC inhibitors, you would have heard about them during your pharmacology days. They are nothing but histone deacylase inhibitors. These molecules are being used in the treatment of squamous cell carcinoma of skin. They are used in the treatment of basal cell carcinoma. These molecules are basically functioning, or they inhibit the deacylation. Hypomethylation makes the cell proliferate. Deacetylation makes the cell proliferate. So when you inhibit both of them, the cells become senescent and they die. This is a concept of using histone deacylase inhibitors for treatment of squamous cell and basal cell carcinoma. I will be discussing about the same epigenetic changes during my colon cancer discussion as well as cutaneous malignancy discussion. Okay. There's a question about loss of function is seen in well-differentiated or poorly differentiated tumor. Whenever there is a well-differentiated tumor, it will resemble anatomically and physiologically to the normal cell, meaning there will be some element of function. If not for 100%, there will be 60 to 70% functioning. Say, for example, a well-differentiated adenocarcinoma of cervix will be secreting your normal mucin, which is secreted by the cervical column. That is what is responsible for your leukorrhea. On the contrary, your poorly differentiated tumors will not have the normal cellular counterpart function. They will not be secreting. They will be madlessly multiplying, and because of demand and mismatch of supply, the cells will undergo necrosis and breakdown. The cells do not break down by themselves alone. They also break down the adjacent blood vessel. That is responsible for bleeding, or clinical presentation as bleeding per rectum or bleeding per urethra, bleeding per vagina in various malignancies. Okay, there is a question about EMTs and METs. EMT, I already told you, whenever there is a cancer cell, it has to get access to spread. The spread can be into local spread or distant metastasis. If it is to the local spread, it will be into your subcutaneous tissues. Distant spread is via your blood vessels or lymphatics. This to access from the epithelial component of cancer. For example, you talk about breast cancer. This is an epithelial component. All the cells here are your ductal and your lobular cells. These cells will be in disguise, or they will get themselves transformed. They will change themselves into your mesenchymal cells for enabling themselves into access into the blood vessels and the lymphatic vessels. When they get access into the lymphatic vessels and the blood vessels, they can easily be transported as a normal mesenchymal element, and once they reach the site or the harboring site of area of metastasis or area of deposition or area of nodal spread, they will again undergo what is called mesenchymal to epithelial transition, where from the blood vessel the cells will be, which is a mesenchymal cell, will get themselves rechanged or transform themselves again into an epithelial component so that they will be permanently deposited. The epithelial cell is stationary. The mesenchymal cells are mobile. So for an epithelial cell to get access to a different site, it has to change itself into a mobile counterpart. This is what in short is referred to as EMT, epithelial to mesenchymal transition. Similarly, a mobile cell, when it has to be identified and deposited into a particular organ of interest, it has to get transformed into a stationary epithelial cell. This is called mesenchymal to epithelial transition. Okay, there's a question like ulcerated lesions and tumors show increased Warburg effect. No, it is not like that. Warburg effect is what is happening within your cellular component. It does not mean that the cells are being destroyed or there is a demand or mismatch. What signifies ulceration or what signifies bleeding or what signifies any tissue disruption is a mismatch between your demand and supply. Warburg effect is even when there is adequate amount of oxygen. That is why I'm mentioning it is not anaerobic glycolysis, it is aerobic glycolysis. Even when there is adequate amount of oxygen, a normal cell, as I told you, will convert your glucose into pyruvate and send them to citric acid cycle, and then finally end product will be carbon dioxide. Whereas the same cancer cell in the presence of same oxygen metabolizes glucose into pyruvate and converts the pyruvate into lactate, because lactate will act as a substrate for the adjacent cancer cells as a food. It will be taken inside by their citric acid cycle as an intermediate substrate, and they will be subjected to production of carbon dioxide and further energy production. The ATPs are required for various cellular metabolism. The cancer cells will require a lot of ATPs because they undergo constant genetic mutations and proliferation. So bleeding and an ulceration signifies poor supply and there is increased demand. Whereas Warburg effect, even though when they have got oxygen, they do not undergo a normal sequence of event and they subject the cells into an alterated pathway where there's a good supply still. There's no poor supply. There's a good supply, but still they assume that they are having poor supply and subject these substrates into your neighboring cells for enabling them to use them. Okay, some other examples of cancers which require surgery as a primary modality of treatment, like your LCAMs. Yes, there are few malignancies like your well-differentiated tumors, your gastric GIST tumors. GIST tumors are usually well-differentiated. The gastrointestinal tumoral tumors, your renal cell carcinomas, your ovarian malignancies, your well-differentiated ones will not have much response to chemotherapy. They will have an higher response to your routine surgical primary modality of management, which will be a sole modality of management as well. Okay, DNA mismatch repair genes are a type of tumor suppressor genes. Yes, they are tumor suppressor genes. They are normally responsible for your cellular multiplication, proliferation, and the cellular events that occurs before transforming into a cancerous tissue. This DNA mismatch repair genes are implicated for all your HNPCC and related mutations, where there will be a absence of control of cell proliferation. So these mismatch repair genes are normal gatekeeper genes or tumor suppressor genes. When they are mutated, the cells undergo uncontrolled proliferation. I think I've taken almost all questions. Any specific questions that somebody wants to ask can contact me through my mobile number. Good. Thank you. Any further queries, you can contact. Okay. Thank you. Bye.